Hello and welcome to Digital Medboard. Here we go. We are examining a blood collection tube with a red cap. A helpful mnemonic for remembering the red top tube is RSS Red, Silica and Serum. The additive used in the red top tube is silica, which acts as a clot activator, helping in serum separation. If you collect a blood sample in a tube containing an anticoagulant, such as EDTA, and the tube cap is lavender in color, this tube is used for plasma separation. Plasma differs from serum. In plasma, it contains all clotting factors, as the blood does not clot. After centrifugation of non-clotted blood, the supernatant that separates is called plasma. On the other hand, if you collect a blood sample in a plain tube without anticoagulants, the negative charges of the tube stimulates in vitro clotting. After the blood clots, the liquid portion that separates is called serum. Unlike plasma, serum does not contain clotting factors such as fibrinogen. We got a question. The additive that is used in the given blood collection tube is the red top tube contains silica, which is used for serum separation and all biochemical analysis, including electrolytes, renal function tests, liver function tests, thyroid function tests, hormonal assays. Even in emergencies, waiting for natural clotting can be time-consuming. Clot activators like silica are added to accelerate the process. And as we already know the answer to this question, option D, silica, is the correct answer. Important points. For biochemical analysis, such as potassium estimation, using an EDTA tube is not recommended because the EDTA additive increases the potassium concentration, leading to falsely elevated results. Similarly, if you use an EDTA tube for calcium estimation, the result will be falsely low because EDTA binds to calcium. EDTA also interferes with many other biochemical parameters. Therefore, for biochemical tests, blood samples should never be collected in EDTA tubes. Instead, serum sample tube should be used. For tests like complete blood count (CBC) or HbA1c glycated hemoglobin, intact RBCs are required. In these cases, a plasma sample is used because it is not clotted, which allows proper mixing and analysis of the sample to obtain whole blood. On the other hand, serum, however, is already clotted, making it unsuitable for the tests. If you found this video helpful, don't forget to like, share and subscribe to Digital Medboard. Got questions? Drop them in the comments below. See you in the next video. Until then, stay tuned. Choose.